which is going to be the basics of DNA and cell division. We'll talk about chromosomes. We'll start talking about genetics a little bit, Punnett squares, all that good stuff. So this is the basics of DNA and cell division, which will correlate with chapter 3 in the explorations textbook. All right, so first let's talk about the types of cells. Okay, so this is going to be different from classes. The types of cells that exist on the planet are prokaryotic cells and eukaryotic cells. Okay, so the vast majority of life on this earth that we're going to be talking about during this class is eukaryotic organisms. These obviously this is a class on humans, and humans are multicellular eukaryotic organisms. But just so you know, there are single-celled organisms on the planet, bacteria. These are no nuclear membranes or organelles. So essentially they are cells that do not have any compartments, any organs. I'll show you a picture of what the organs look like here in a moment. But they're single-celled organisms. They have very simple single-stranded DNA that is completely contained within the cytoplasm. So the cytoplasm is the liquid that kind of helps keep the cell's shape. Prokaryotic organisms, bacteria, are believed to be the first life on Earth. And it's believed that the first prokaryotic organisms originated about 3.5 billion years ago. The planet itself, like we learned last lecture, according to geology and paleontology, the planet itself is around 4.6 billion years old, but we see evidence for the first prokaryotic organisms about 3.5 billion years ago. Okay, so the vast majority of the class, of course, we're going to be focusing on the second type, which is eukaryotic or multicelled organisms. So multicelled organisms do, in fact, have a membrane-bound nucleus. So you can think of the nucleus as kind of the brain of the cell. We'll look at a picture here in a moment, where the nucleus is the brain of the cell. Eukaryotic organisms do, in fact, have double-stranded DNA. And there's two different types of DNA that we'll talk about here in this lecture. We'll talk about the nuclear DNA. So the nuclear DNA is the actual genetic material that makes up our 46 chromosomes. So DNA coils around these proteins called histones to form visible chromosomes. And I'll show you a picture of what that looks like here in a moment. But for now, know that there's two different types of DNA. There's nuclear DNA contained in the nucleus. And then the second type is mitochondrial DNA. So mitochondrial DNA, you might guess, is contained within the mitochondria. The mitochondria is an organelle within the cell that is the powerhouse of the cell. I'm sure you guys have heard this one before. But it's not only responsible for creating the gasoline or the ATP for the cell, but it also holds the mitochondrial DNA, which, even though it's not the actual genetic material, mitochondrial DNA is fascinating in the sense that it's only inherited from your mother. So when you're looking at tracing ancestry and tracing migration routes and tracing the matriline, the mother's line, mitochondrial DNA is exceptionally useful. All right, so eukaryotic organisms do, in fact, have specialized structures called organelles. So you can think of organelles as the compartments or the organs of the cell. I'll show you a picture here in just a moment. And we see evidence for the first eukaryotic life going back about 1.2 billion years ago. Okay, so this is that picture of the eukaryotic cell I was promising you. So for this class, there's really only 3 organelles that I really want you to focus in on. Okay, I want you to focus in on the nucleus, the mitochondria, and the ribosomes. And you'll understand why as we go through the lecture. But just for now, know that the nucleus is kind of the brain of the cell. It houses nearly all of the genetic material or DNA for that organism. So all of it, for humans, for example, the nucleus contains all 46 of our chromosomes that code for all of our genetic components. It's covered by a nuclear membrane or nuclear envelope, which keeps the contents of the nucleus separate from the rest of the cell. So it's that big purple mass in the center here. And then all of that squiggly stuff looks like spaghetti. That's the DNA in there. Those are the chromosomes all mixed up. All right, the second organelle I want you to be familiar with is the red oval shapes. Those are the mitochondrions. So the mitochondrions are the powerhouse of the cell because it generates most of the energy. So think about the mitochondria as like the gas tank that holds all the gasoline. So the number of mitochondria per cell will vary by the tissue type and by the organism. And the other reason the mitochondria is important for us here in an anthropology class is it also houses not just the energy, but also the mitochondrial DNA, which for us, that's important because it helps us trace the matriline or the mother's line, because we only inherit mitochondrial DNA from our mother. All right, the other organelle that will be important as we go through this class and this lecture is the ribosomes. Those are actually the little blue dots that you see on the endoplasmic reticulum. Those little blue dots are the readers. So when we start learning about protein synthesis, which is the way that our genes are read, those little blue dots, the ribosomes, will read the proteins, will read the codons to determine what protein is being coded for. So without those little ribosomes, we wouldn't be able to read our genetic code, or our cells wouldn't be able to read our genetic code. So that's why we care about them. Okay, so classes of cells, a little bit different than types of cells. So now we're just exclusively focusing in on eukaryotic. So you can not completely forget about the prokaryotes, but kind of, put those out of sight, out of mind for now. Because the vast majority of the semester, of course, we're going to be focusing in on humans, which are multicellular eukaryotic organisms. So within a human, for example, we have two classes of cells. The vast majority of the cells within our body are called somatic cells, also known as body cells. So somatic cells make up all of the cells within our body. So our tissues, our organs, our nerves, our bones, our blood, our brain, et cetera, et cetera. Basically every cell within our body that's not a sex cell. So if it's not an egg cell and it's not a sperm cell, it's a somatic cell. So somatic cells are what we call diploid. So think diploid double, haploid half, diploid double, haploid half. So within a human, a typical human, a diploid cell is going to contain every chromosome that we need. So every single skin cell, every single organ cell, every single blood cell within the nucleus of those cells contains 46 chromosomes in humans. So that's the diploid number for humans, is 46, okay? 46 individual chromosomes or 23 pairs. Okay, so that's why when you're watching forensics files and they are able to extract a saliva cell or a blood cell or even some skin cells, if they have the proper DNA technology and the DNA hasn't been contaminated, then they can actually determine who that person is based upon that, just a few cells, because DNA technology allows us to do that. Okay, so it's important to remember that every single cell within our body, every skin cell, organ cell, tissue cell, bone cell, contains an entire set of our chromosomes, all 46. All right, the other class of cell that we find within humans is going to be gametes. So the other way of thinking of a gamete is a sex cell, an egg cell, or a sperm cell. So gametes are what we call haploid. So remember, diploid, double, haploid, half. Diploid, double, haploid, half. So half of 46 is what? Half of 46 is 23. So every single sex cell, every egg cell, every sperm cell is haploid. So it only contains half of our diploid number. So 23 chromosomes. So every egg cell is 23 chromosomes in the nucleus. Every sperm cell is 23 chromosomes in the nucleus. Okay, so once those two combine, once the sperm cell and the egg cell combine and they are fertilized, they become a zygote. Okay, so quite literally, genetically speaking, every single one of us is 50% mom, 50% dad. Now, of course, some genes are dominant over others. Some genes, some genotypes are influenced by the environment. But in general, we all are 50% mom, 50% dad, genetically speaking. Okay, so 23 of our chromosomes are going to be obtained from our mother's ovum or egg cell. And then the other 23 chromosomes are obtained from our father's sperm cell. Okay, so the combination of that will make up our genome and eventually our genotype that we'll learn about today as well. Okay, so a zygote is simply a fertilized egg that is also, once it becomes a zygote, it's also diploid because it has 23 chromosomes from mom, 23 chromosomes from dad. And that fertilized egg will travel down the fallopian tubes and eventually attach to the uterine wall and eventually become a developing fetus. Okay, but it starts out as a zygote. All right, questions so far? Nothing. All right, so this is just a picture, just an example of what a somatic cell might look like. This one's an osteocyte or a bone cell. All right, let's talk a little bit about DNA. So DNA is exceptionally important because it is the blueprint of life. It is the chemical template for every aspect of an organism, okay? So in a eukaryotic organism like a human, the DNA is in a double helix ladder-like structure, okay? So it looks kind of like this image at the bottom of the slide here. So it's like a ladder that's all twisted up, okay? So the rungs of the ladder, so that what you would step on, oops, the rungs of the ladder are made up of these alternating nitrogen base pairs, okay? So in DNA, the rungs of the ladder are made up of these alternating complementary nitrogen base pairs. So if it's DNA, adenine will always pair with thymine. Okay, and it's just a memorization here. Adenine will always pair with thymine, and guanine will always pair with cytosine. So if it's DNA, it's always A to T and always C to G. Okay, now it'll be a little bit different once we start learning about RNA, but to avoid confusion, let's focus first on just DNA. So if it's DNA, adenine will always pair with thymine, and guanine will always pair with cytosine every single time. Okay, so that's the rungs of the ladder. So the building blocks are the nucleic acids. These are called nucleotides. So each nucleotide has three components. So the first component will be a sugar molecule. The second component will be a phosphate molecule. And the third component will be one of those nitrogen base pairs, either adenine, thymine, guanine, or cytosine. Okay, so the sides of the ladder, that's what's made up of the alternating sugar and phosphate molecules. The rungs of the ladder is made up by the alternating complementary nitrogen base pairs. Okay, so the type of sugar determines whether it's a DNA molecule or an RNA molecule. So DNA has a type of sugar that's called deoxyribose. So that's where we get the D in DNA. Okay, so if it's DNA, the sugar molecule is called deoxyribose, which is where we get the D in DNA. If it's RNA, it's called ribose. So that's where we get the R in RNA. Okay, so the sugar molecule type determines whether it's DNA or RNA. Okay, so the rungs of the ladder are made up by these alternating nitrogen base pairs. Adenine always pairing with thymine, guanine always pairing with cytosine, and vice versa. Okay, so three components, sugar molecule. If it's DNA, it's called deoxyribose. If it's RNA, it's called ribose. Second component is the phosphate molecule. Third component is the nitrogen base. Adenine pairing with thymine and guanine pairing with cytosine. I'm repeating and doing lots of repeating, guys, just because there's a lot of memorization in this chapter. So, at least when I'm learning, I find sometimes it's helpful to hear things over and over again. But if I get too excessive with it, please feel to let me know. Okay. I'm just doing it to help your memorization. Okay, so the two types of DNA. So I mentioned in eukaryotic organisms, there's two types of DNA. There's the nuclear DNA, which is mainly what we'll be focusing on. Nuclear DNA is the actual genetic material that makes up our 46 chromosomes. So DNA will coil around proteins called histones to make these visible chromosomes that are contained within the nucleus of all of our somatic cells. Okay, so nuclear DNA is contained within the nucleus. That's easy to remember, right? Nuclear DNA in the nucleus. It makes up our 46 chromosomes. A complete set, so within every somatic cell, we have a complete set of our nuclear DNA, all 46 chromosomes, and we call that a genome. So a genome is just one organism's, one person's DNA. So for humans, it means all 46 chromosomes. So when you hear about mapping the genome, that's what they mean. They're looking at all 46 chromosomes, all of those nitrogen base pairs, all of those genes. And that's what they mean by mapping the genome. It's just one person's genes. Okay. Now, a gene pool, which we'll learn about in a future chapter, a gene pool means all of the genomes of all of the people in that population. So if we were to all migrate to an island, and let's say that there's 40 of us, a genome would be just one of ours one of just my genetic material, for example. But a gene pool would be all of our genes combined together. Okay. Nuclear DNA is homoplasmic, so break that word up. The word homo means same, plasmic is referring to the DNA, the plasma. So it refers to nuclear DNA, which is identical in the nucleus of each cell type. So within every single skin cell, every skin cell's DNA within our cells is identical to our blood cells, to our bone cells, to our nerve cells. Now, of course, there are, cells specialized, but essentially every single one of our cells, all of our somatic cells at least, contains a complete set of our instructions. All right, so that's what the nuclear DNA looks like underneath a microscope like that. Okay. All right, so the only other type of DNA within a eukaryotic organism is called mitochondrial DNA. Okay, so just like it sounds, mitochondrial DNA is contained within the mitochondria. It's a little bit in the cytoplasm, but it's mainly within the mitochondria. And you may see it abbreviated at lowercase mt and then uppercase DNA. So just know if you see that in the reading or in a PowerPoint slide or in a film, just know if it's mtDNA, they're just abbreviating mitochondrial DNA. Okay, so mitochondrial DNA is contained within the mitochondria of a eukaryotic cell, and it's inherited exclusively from the mother. Okay, so you may, when you're reading a DNA study, if they're talking about mitochondrial DNA, that means that they're likely interested in the matriline. They're interested in tracing that organism's maternal lineage, or mother to grandmother to great grandmother, so on and so forth. Okay, so in human evolution, we'll learn about some studies that have traced the matriline back hundreds of thousands of years. So that search for the mitochondrial Eve, which is kind of ironic because the term Adam and Eve is definitely a religious connotation. So it's kind of ironic that, and a lot of studies we read, we're searching for that mitochondrial Eve. All right, so DNA is going to be responsible for two vital processes that we'll learn here in this lecture. Okay, so the first one is going to be replication. The second one will be protein synthesis. Okay, so don't worry, it might seem a little bit complicated at first, but I promise we're going to break it down and try to make it as easy to understand as possible. Okay, DNA replication is pretty easy. Protein synthesis gets a little bit more complicated, but we'll break it down and make it super easy to understand. Okay. So this slide just lists the two processes, and we'll talk about it in more detail. So the two processes that we care about with DNA for right now is replication and protein synthesis. All right, so DNA replication will occur entirely in the nucleus of the cell. It goes down entirely in the nucleus, and it will occur during the prep phase or pregame phase of both mitosis and meiosis. Okay, so we'll learn about mitosis and meiosis in this lecture too. But for now, just know that it happens as the cells are preparing to divide. Okay, so mitosis is when the somatic cells or the body cells divide, and then meiosis is the process that makes genetically unique gametes, sex cells. Okay, so DNA replication is necessary prior to mitosis because remember, every cell needs an identical copy of our genetic code of all 46 chromosomes. Okay, so the cell must go through this process of DNA replication in order to prepare for cell division. Okay, so during this process of DNA replication, the double helix will essentially unzip and expose the base pairs of the parent strand. Okay, so those base pairs will be exposed. So all of the adenines, all of the thymines, all the cytosines, all the guanines are now exposed. So in this picture on the right-hand side, you see this blue ribbon, and you see that blue ribbon kind of unzipping. Okay, so as it unzips, it exposes these nitrogen base pairs. And then the yellow or gold colored ribbon, that's representing the new or the daughter strand. Okay, so the blue strand, that's the parent strand or the old strand. The yellow strand, that's the new strand or the daughter strand. Okay, so as these nitrogen base pairs are exposed, all the adenines, all the thymines, all the cytosines, and all the guanines, this new strand, the daughter strand, comes in and pairs up following those rules we learned about. So that's why we need to know that adenine will always pair with thymine and cytosine will always pair with guanine every single time. If it's DNA, if we're talking DNA replication, every single time adenine will pair with thymine and cytosine will pair with guanine. Every single time, no exceptions. Always A to T, always C to G. Okay, so this process is what we call semi-conservative. So semi-conservative meaning that at the end, see we have these two strands at the end, and each of them are half blue and half yellow. Remember, the blue strand is representing the original or the parent strand, and the yellow ribbon is representing the new or the daughter strand. Okay, so semi-conservative process, meaning that at the end we have two identical strands of DNA, and each is half the old and half the new. So it's semi-conservative. All right, so now we're going to watch a little clip from Amoeba Sisters. Any questions about DNA replication before we watch the clip?
No, thank you, doctor.
Excellent. Okay. All right, so let's go here to DNA replication. So I like these little Amoeba Sister kind of animations of DNA replication and the other processes we're going to learn. So they can be a little bit, they're animated, and they can be a little bit, they can seem a little bit silly at first, but actually they really are very accurate and break down the processes really, really well. It keeps it really simple. And in an intro class like this, I really just want you guys to know the basics. So, as I encourage you, as you're watching this, don't feel the need to rigorously take down notes. I mean, of course, these are all in the modules if you want to watch them again. The best thing you can do right now, I think, is just kind of sit back and watch the animation. And of course, if you have any burning questions, definitely jot it down so you don't forget to ask it once the film clip ends. But I want you to mainly focus on just watching this animation to understand how this process would work. Okay? Sound like a fair deal?
Yes.
Let me share one more time just to make sure I optimized for sound. Here we go. All right, so let's watch this and we'll talk about it or any questions you might have.
DNA. We talk about it so much. It's the ultimate director for cells, and DNA. We talk about it so much. It's the ultimate director for cells, and it codes for your traits. With a molecule that has a function like that, it makes sense that when you make another cell, like in cell division, you would also need to get more DNA into the new daughter cell. And that introduces our topic of DNA replication, which means making more DNA. First, let's talk about where and when. First, where? Well, if it's in a eukaryotic cell, it occurs in the nucleus. However, remember, not all cells have a nucleus, such as prokaryotic cells. They don't have a nucleus. Still, both prokaryotic and eukaryotic cells do DNA replication, but there's some differences between the two that this clip doesn't go into. Next, when? When does this happen? Well, a cell is going to need to do this before it divides so that the new daughter cell can also get a copy of DNA. To get specific, in a eukaryotic cell, that's going to be before mitosis or meiosis in a time known as interphase. I think DNA replication would actually make a great video game. Still waiting for that to be invented. I'm going to introduce the key players in DNA replication so that you can get some background information. Now remember, these are just some major key players. There's a lot to this process. Many of the key players are enzymes. In biology, when you see something end in ASE, you might want to check, as it's very possible that it's an enzyme. Enzymes have the ability to speed up reactions and build up or break down the items that they act upon. So here we go with the key players. Helicase, the unzipping enzyme. If you recall that DNA has two strands, you could think of helicase unzipping the two strands of DNA. Helicase doesn't have a hard time doing that. When unzipping, it breaks through the hydrogen bonds that hold the DNA bases together. DNA polymerase, the builder. This enzyme replicates DNA molecules to actually build a new strand of DNA. Primase, the initializer. With as great as DNA polymerase is, DNA polymerase can't figure out where to get started without something called a primer. Primase makes the primer so that DNA polymerase can figure out where to go to start to work. You know what's kind of interesting about the primer it makes? The primer is actually made of RNA. Ligase, the gluer. It helps glue DNA fragments together. More about why you would need that later on. Now, don't feel overwhelmed. We'll go through the basics of this sequence in order. But remember, like all of our videos, we tend to give the big picture. There are always more details and exceptions to every biological process that we can't include in such a short video. DNA replication starts at a certain part called the origin. Usually this part is identified by certain DNA sequences. At the origin, helicase, the unzipping enzyme, comes in and unwinds the DNA. Here's the thing though, you don't want these strands to come back together. So SSB proteins, which stands for single-stranded binding proteins, bind to the DNA strands to keep them separated. And topoisomerase, I always have to slow down when I say that enzyme's name, keeps the DNA from supercoiling. Supercoiling might sound super, and it can be when you're trying to compact the DNA, but it's something that needs to be controlled during DNA replication. Supercoiling can involve an overwinding of the DNA, and you need the DNA strands to be separated for the next steps. Primase comes in and makes RNA primers on both strands. This is really important because otherwise DNA polymerase won't know where to start. In comes DNA polymerase. Okay, before we go on, remember how we said DNA has two strands? They're not identical, they complement each other. In our video that covers DNA structure, we talk about how the bases pair together with hydrogen bonds. The base adenine goes with the base thymine, and the base guanine goes with the base cytosine. These strands are also anti-parallel, so they don't go in the same direction. What do we mean by direction? Well, with DNA, we don't say north or south. We say DNA either goes 5' to 3' or 3' to 5'. What in the world does that mean? Well, the sugar of DNA is part of the backbone of DNA. It has carbons. The carbons on the sugar are numbered right after the oxygen in a clockwise direction. 1', 2', 3', 4', and 5'. The 5' carbon is actually outside of this ring structure. Now, you do the same thing for the other side, but keep in mind DNA strands are anti-parallel to each other. So let's count these, again clockwise after the oxygen. 1', 2', 3', 4', 5'. And the 5' is out of this ring. This strand on the left runs 5' to 3', and the strand here on the right runs 3' to 5'. We'll explain why all that matters in a moment. So let's take that knowledge there and look at DNA replication here. In this image, I labeled the top original strand 3' to 5'. I labeled this bottom original strand 5' to 3'. That's the original DNA that is going to be replicated. DNA is unwinding here thanks to helicase. In this example, it will keep unwinding in this direction. Primase places primers. DNA polymerase is building the new strands. Now, the thing about DNA polymerase is, when it's building a new strand, it can only build the new strand in the 5' to 3' direction, meaning it adds new bases to the 3' end on the new strand. See how it's being built in the 5' to 3' direction? This one is called the leading strand. But take a look down here. So DNA polymerase once again is building a new strand in the 5' to 3' direction. But there's a bit of a problem here. See, as DNA unwinds, because DNA polymerase can only build the new strand in the 5' to 3' direction, it has to keep racing up here next to where this unwinding is happening. You can see why then this new strand is known as the lagging strand. On this lagging strand, primers have to keep being placed in order for DNA polymerase to build. These fragments that result are known as Okazaki fragments. Primers have to get replaced with DNA bases since the primers were made of RNA. Ligase, the gluing enzyme, as I like to nickname it, has to take care of the gaps between Okazaki fragments, sealing them together. At the end of replicating, you have two identical double helix DNA molecules from your one original double helix DNA molecule. We call it semi-conservative because the two copies each contain one old original strand and one newly made one. One last thing. Surely you've had to proofread your work before to catch errors. In this process, you don't want DNA polymerase to make errors. If it matches the wrong DNA bases, then you could have an incorrectly coded gene, which could ultimately end up in an incorrect protein, or no protein. DNA polymerase is awesome. It has proofreading ability, meaning it rarely makes a mistake, which is a good thing. So remember how we said there is far more detail to this process to explore? The detailed understanding of DNA replication has led to some life-saving medical treatments that can stop DNA replication in harmful cells, including pathogenic bacteria or human cancer cells. We encourage you to explore beyond the basics. Check out the further reading suggestions and the video details to explore more. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
All right. So I know there was a lot of details in that video, but really, really what I want you guys to understand is the basics. I want you to understand that, that yes, DNA helicase inspires that parent strand to unzip. So remember, anytime it's an ACE, it's an enzyme, and enzymes inspire molecules to do things. So the DNA helicase inspires that original parent molecule, that parent strand, excuse me, to unzip, which is then exposing all of those nitrogen base pairs, all the adenines, all the thymines, all the cytosines, and all the guanines. Okay, so then that daughter strand comes in 5' to 3', and then the adenine pairs with the thymine, and then the cytosine pairs with the guanine. And then your end result, why we care, the end result, we've got two identical strands that are both, they're semi-conservative. Each strand is half the parent strand and half the daughter strand, half the old, half the new. So half blue, half yellow in the photo here. All right, what questions do we have, if any?
What causes them to grow and to expand? What spark is causing all these things to replicate, duplicate, and grow into the organism? Is there
So it's necessary for life to happen. So without cell division, we would die, right? If our cells couldn't repair themselves, we would die real quick, right? So this is a process that has to occur in order for each somatic cell to have a complete copy of our DNA. So it's the questions you're asking, I guess, are ones I don't have answers to, but without this, we wouldn't be able to survive. We would die.
Sure. Thank
right?
you.
You're welcome. Good question. A good question. I don't think I necessarily have the correct answer that you're searching for, but a good way to think about it is that, without DNA replication, we couldn't have cell division. And then without cell division, if we got a scrape on our arm, that scrape on our arm would never be repaired. Or if we had some damage to our organs, we would never be able to repair our organs. Or if we had a bone break, if we fractured our femur, we would never be able to repair that femur. So it's, without DNA replication, we wouldn't have cell division. Without cell division, we wouldn't be able to repair our cells as they become damaged. And, that would mean we would have a very short lifespan.
And
Good question.
My thought would be is simple. Who's hitting the switch? What's the switch that gets this in motion? And we can talk
the
about
helicase. Yeah, the helicase. The DNA helicase is the switch.
Got it. Perfect. Thank you.
Does that help? Yes.
Yes.
Awesome. Good question. Other questions? Nothing? Okay. All right, so let's move on to our next process. So this is the next process that is vitally important that DNA is responsible for. Okay, so just to review, when we're talking DNA, the DNA replication, adenine will pair with thymine, guanine pairs with cytosine, thymine pairs with adenine, and cytosine pairs with guanine. That's not new information. Now, when we start learning about protein synthesis, RNA is going to come into play. It's going to come on the scene. So the only difference when we're talking RNA, the only difference is adenine is now going to pair with a new nitrogen base. It's called uracil. Okay, so that's the only difference. Everything else is the same. And when we're talking RNA, adenine will now pair with the nitrogen base called uracil. Okay, and you'll understand why this is important here in a moment. Okay. All right, so let's talk protein synthesis. So proteins are essentially the chemicals that make up our bodily tissues. Okay, so proteins essentially make up our genes. Okay, those are the components of our DNA that codes for our physical attributes, that are essentially our genes, our genetic material, our genetic code. So proteins also function, they also regulate functions, bodily functions, they repair, and they also allow for the growth of our bodily tissues. So they regulate, they repair, and they allow us to grow. All right, so proteins are made up of 20 different amino acids. So amino acids are organic molecules that will combine in a specific sequence by the ribosomes to form a protein. So there's that organelle coming up again, the ribosomes. That's that third organelle that I wanted you guys to be aware of in addition to the nucleus and the mitochondria. Okay, so we'll learn how the ribosomes read the code here in a moment. But for now, know that amino acid is an organic molecule that combines in a specific sequence by the ribosomes to form the protein. So a protein is made up of a sequence of amino acids, essentially, or multiple amino acids. A chain of amino acids, you might say. All right, so within our diet, we do essentially, within our body, we have amino acids that just occur naturally. And some of those amino acids we need to get from our diet. So that's why sometimes when you have a vitamin deficiency, it can actually have really catastrophic health consequences, okay? Because our body is capable of producing essential amino acids, but some of them we may need from our diet in order to completely function properly. All right, so just like there are types of cells and types of DNA, there are also different types of proteins. So the two types that we'll talk about here in this class are structural proteins and regulatory proteins. So structural proteins, just like they sound, are responsible for our bodily structures, our physical characteristics, okay? So an example of a structural protein would be keratin and collagen that's found in our skin and in our nails. Okay, those are examples of structural proteins. The second type is a regulatory protein. So just like it sounds, regulatory proteins are responsible for bodily functions. So things like enzymes, hormones, and antibodies, those are all examples of regulatory proteins. Okay. Protein synthesis, and we're going to learn about it in detail here in a moment, but just know for now it involves two major steps. The first step being transcription, which transcription is going to sound a lot like DNA replication. It's going to start just like DNA replication. It involves the unzipping of a parent strand to make a template for RNA, okay? So protein synthesis starts with transcription, which is in the nucleus of the cell. And then that RNA is actually going to be single cell or single stranded, excuse me. That RNA is single stranded, which means it's small enough to leave the nucleus and travel out into the cytoplasm where the ribosomes are located, okay? So that's the big difference is DNA replication happened entirely in the nucleus of the cell. Protein synthesis will start in the nucleus and then it's going to move out into the cytoplasm because it's going to need the ribosomes to read the code. Okay, so first step, transcription, occurs entirely in the nucleus where the DNA resides, and it will result in the formation of a messenger RNA strand. Okay, so the DNA strand unzips, so it starts just like DNA replication. The parent strand unzips and exposes all those nitrogen base pairs. Okay, so the biggest difference is when we're talking protein synthesis, we're going to have this messenger RNA strand. So instead of adenine pairing with thymine, adenine will now pair with uracil. Okay, so that messenger RNA strand is now small enough to leave the nucleus and travel out into the cytoplasm, carrying that message. So that's going to be step 2. The second step is called translation. Okay, so during translation, that messenger RNA is single-stranded. It leaves the nucleus and carries the message or the genetic code out into the cytoplasm. Okay, so once it's out in the cytoplasm, the codons are going to be read by the ribosomes. Okay, so the ribosomes, those are those little blue dots, those little blue organelles that are located on the endoplasmic reticulum. So those little ribosomes, they read or translate the genetic code into a specific order of amino acids. Okay, so there's going to be an amino acid chart that we'll see here in a moment in the little video clip we're going to watch. But the amino acid chart is going to read codon by codon. So a codon is a set of 3. Okay. So the two processes look right next to each other. So this is transcription and translation right next to each other. So transcription occurs entirely in the nucleus of the cell. It starts out just like DNA replication. It involves DNA helicase inspiring the parent strand to unzip. And then it's making this complementary copy or blueprint of DNA. Okay, so that's the DNA template. Okay, so the DNA template is what the messenger RNA strand will use to pair up with. Okay, so Now remember, when we're talking RNA, anytime you see RNA instead of DNA, just remember that adenine will now pair with uracil, okay? So the reason that we need 2 steps is because we need to go out into the cytoplasm where the ribosomes are located so that they can do the reading. Okay, so messenger RNA is small enough to leave the nucleus and travel out into the cytoplasm to get those ribosomes. Okay, all right, now step 2 is called translation. So translation is going to occur entirely in the cytoplasm of the cell. So that messenger RNA carries its message of sequences of DNA bases, and the ribosomes are going to read the messenger RNA 3 at a time. Okay, so it's 3 nitrogen base pairs at a time. So we call a set of 3 a codon. So an A, U, and a G, for example, is a start codon. AUG, adenine, uracil, guanine. That's the start codon. That's the switch that we were talking about. Okay, so ribosomes read the messenger RNA bases 3 at a time, codon by codon. And then there's another type of RNA called transfer RNA. So transfer RNA is going to bring those amino acids into place and lock them in there. Okay, so the AUG is the start codon, for example. So once those amino acids are brought in, eventually we're going to have a chain of amino acids, and we call that a polypeptide chain. So break that word up again. Poly means many, peptide means protein. Okay, so that messenger strand of RNA, the ribosomes are reading it codon by codon, and they're locking in these chains of amino acids. And this will make more sense, I think, once you can see it in the animation. So end result, you've got this chain of amino acids, polypeptide chain that codes for a protein, okay? So the reason we care, the reason we're even learning the basics of this process is this is what codes for our genes or our genetic makeup, okay? All right, so let's watch the animation. So it's another amoeba sister one. So this one is going to really show you how this works. Okay, so remember, don't worry about knowing every single detail. I want you to focus on the bigger picture here. Focus on the bigger picture and the end result of the process.
After learning about DNA, have you ever wondered how can the DNA actually result in a trait? Let's take an example, like eye color. Yes, your DNA has the genetic information that codes for the color of your eyes. Your eye color is based on a pigment that is inside the eyes, but in order to have that pigment, you have genes, which are portions of DNA that can code for proteins which help make that pigment. So what we're going to talk about is how your DNA can lead to the making of a protein. This process is called protein synthesis. Synthesis essentially means to make something, so protein synthesis means to make protein. And you may wonder, what's a big deal about proteins? Well, you may not realize this, but proteins are kind of a big deal. They do all kinds of things. Proteins are involved in transport, in structure, in acting as enzymes that make all kinds of materials, in protecting the body, and so much more. You've got to make proteins. It's essential for you to live. And what is so cool is that you are making proteins right now as you sit and watch this video. It's happening in your cells. They're making proteins. So back to your DNA and its role in all of this. All of your cells have DNA. Well, a few exceptions. And that DNA is in the nucleus. Some DNA is non-coding DNA. Some DNA makes up genes that are not activated. More about that in our gene regulation video, but we're going to talk about genes that are coding for active proteins. So how are we going to get the information from these genes out of the nucleus so that the cell can start producing the proteins that it needs to make? Well, let us introduce you to the amazing work of RNA. We have a video comparing and contrasting RNA and DNA. We'll be brief here in saying that RNA is a nucleic acid, like DNA, but it has a few differences. Its role in protein synthesis also is huge. Before we get into the process, please note our typical disclaimer. We tend to simplify topics, but as always, we hope that you have the desire to explore this complex, amazing process later on, to learn all about the extra information that we don't have the ability to include in this short video. In protein synthesis, we can look at two major steps. One is transcription and the other is translation. Transcription has a C in it, and translation has an L in it. I remember that C comes before L in the alphabet, which helps me remember that transcription comes first. I like a lot of alphabet mnemonics. Now, transcription is when we're going to transcribe the DNA into a message. In your cells, the DNA is in the nucleus, so therefore we're doing transcription in the nucleus. In the step of transcription, an enzyme called RNA polymerase will connect complementary RNA bases to the DNA. These RNA bases are bonded together to form a single-stranded mRNA. The M in mRNA stands for messenger. Messenger RNA consists of a message made of RNA that has been based on the DNA. We do want to mention that this mRNA is not usually ready to go right away. There's usually a significant amount of mRNA editing that occurs. We highly encourage you to do some reading about that because it's not only fascinating, it's critical for the process to work correctly. So what's something great about being mRNA? Well, in eukaryotes, you get out of the nucleus. The mRNA can go out of the nucleus into the cytoplasm where it's going to attach to a ribosome. Ribosomes make protein. The ribosome is made of rRNA, and that's an easy one to remember because the R stands for ribosomal RNA. The ribosome is going to build our protein in the next step called translation. You know, you can find a lot of great clips and animations on translation that are just fantastic. We're just going to break down some basics of what's happening. In the cytoplasm, if you look at this, you have all these tRNA molecules available. tRNA stands for transfer RNA. They carry an amino acid on them. An amino acid is the monomer for a protein. It's a building block for protein. Since we're making proteins, we're going to need those amino acids to build it. If you have a bunch of amino acids together, you can build a protein. So it's the tRNA that is going to bring those amino acids together to make that. But wait, how does the tRNA know which amino acids to bring? That's why the mRNA, the message, is so important because it's going to direct which tRNAs come in and therefore which amino acids are transferred. All of these tRNAs are looking for complementary bases. When they find the complementary bases on the mRNA, they transfer their amino acid. When tRNA is bringing in the amino acids, it reads the bases, represented by these letters here on the mRNA, in threes. So it doesn't read one letter at a time, it reads in triplets. That's called a codon. So for example, in this mRNA, the tRNA would read the codon AUG. One of these tRNAs contains a complementary anticodon, which in this case is UAC. All tRNAs that have the anticodon UAC will be carrying an amino acid called methionine. A tRNA with the UAC anticodon comes in to pair with the complementary AUG codon on the mRNA. It transfers the amino acid it carries, methionine. The tRNA will eventually leave, but it will leave behind its amino acid. That's the first amino acid before looking at the next codon. Before we do the next codon to carry this on, if you're wondering, how did you know that the tRNA that went with the AUG codon would be carrying an amino acid called methionine? Well, for that, you will find a codon chart helpful. You can learn to use a codon chart to determine which amino acid each mRNA codon will code for. Isn't it so fascinating that scientists have been able to determine which amino acid corresponds with these codons? I used to have a codon chart poster and just marvel at that. You could see on a codon chart that the AUG codon on the mRNA codes for methionine. AUG is also considered a start codon, as methionine is typically going to be your first amino acid in proteins. There are many types of amino acids in the codon chart, but there are even more possible codon combinations. That means there can be more than one codon that code for the same amino acid. For example, according to the mRNA codon chart, all of these mRNA codons here code for the same amino acid, leucine. That means their complementary tRNAs all carry the same amino acid, leucine. Okay, so going back to the mRNA, let's try the next codon on this mRNA, CCA. On the codon chart, you can see that codes for the amino acid proline. The complementary tRNA has the anticodon GGU. And look, there's the proline that we knew it would be carrying. the tRNA will transfer that amino acid and eventually leave where it can pick up another amino acid. These amino acids are held together by a peptide bond, and it will keep on growing. Typically, at the very end of the mRNA, there is a stop codon. Stop codons do not code for an amino acid, but when the ribosome reaches it, it indicates that the protein building is finished. So, the result of translation is that you built a chain of amino acids that were brought in certain sequences based on the coding of the mRNA. But remember, that mRNA was complementary to the DNA, so the DNA ultimately was the director of the entire protein building. Of course, it couldn't have done it without some serious help from mRNA, rRNA, and tRNA. Protein folding and modification may occur, and the protein may need to be transported. This can all vary based on the protein structure and function. Another fascinating topic for another Amoeba Sisters video. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
All right. Okay, so what we're going to do in our breakout rooms today, here, let me type it out in the chat box so that we make sure everyone understands. So we're going to actually pair up. There's going to be one group of 3, so we're going to pair up with a partner that will be randomly selected in the breakout rooms. And one of you will describe DNA replication to the best of your ability. Write out in a step format. Okay, so everybody write down what I'm putting in the chat box right now, because I think once you go in the breakout rooms, I think this disappears. So go ahead and write this down or take a screenshot, whatever you want to do. Okay, so everybody write down those instructions, because I think once we go in the breakout room, I think they disappear. So I'm going to go ahead and stop the recording here.